Welcome to my YouTube channel, Perfect Lab. And in today's video, we are going to learn about the Petri dish, which is one of the most commonly used laboratory ware for growing and observing microorganisms in the lab. I will also introduce you to the different types of Petri dishes, explain their key features, and discuss the advantages of each type. So let's get started. A Petri dish can be defined as a shallow, round container made of glass or plastic that is used in microbiology laboratories to grow and culture microorganisms such as bacteria and fungi on culture media like agar. So this is a picture showing the parts of the Petri dish. And the transparent container here is called the base or dish, and it is used to hold the culture medium, which is used to culture these microorganisms in the lab. And the transparent lid here is used to cover the agar medium in the base of the Petri dish. Types of Petri dishes. So there are two common types of Petri dishes based on the material from which they are made. And these are the glass Petri dish, which is made of glass, and the plastic Petri dish made of plastic. And the advantages of glass Petri dishes are: 1. They are reusable after proper cleaning and sterilization. And 2. They are resistant to heat and chemicals. And 3. They are durable and can be used repeatedly. And the advantages of the plastic Petri dishes are: they are lightweight and easy to handle, and most are supplied pre-sterilized and ready to use, and they are convenient for routine microbiological work, and they are mostly disposable to reduce the need for cleaning and sterilization after use. So, apart from the two most common types of Petri dishes that we have other types of Petri dishes that are normally used in the microbial lab. And these are the compartmental Petri dish, the non-vectored Petri dish, and the vented Petri dish. So let's look at the compartmental Petri dish. And this is also called a multi-section or divided Petri dish. And it is a type of Petri dish that contains two or more separate compartments divided by physical barriers. And each compartment can hold a different culture media or microorganisms without mixing with the others. So let's look at the types of compartmental Petri dish we have. So the first is the bi-compartmental Petri. And this is a type of compartmental Petri dish that has two compartments. And we also have the tri-compartmental Petri dish with 3 compartments. And we have the multi-compartmental Petri dish that has either 4 or more compartments. Advantages and the disadvantages of compartmental Petri dish. So let's start by looking at the advantages. So 1, it saves space in the incubator. And 2, it reduces the number of Petri dishes needed. And it also conserves culture media and laboratory materials. And finally, it allows comparison of different microorganisms or growth conditions in the same Petri dish. And for the disadvantages, each compartment has limited space for microbial growth, and it is not suitable when large culture areas are required, and care must always be taken to avoid cross-contamination during incubation when using the compartmental Petri dish. The next is the non-vented Petri dish, and this is a picture of it. And it is a Petri dish whose lid fits tightly over the base without any ventilation gaps or raised ribs. And this is purposely designed to minimize air exchange between the inside of the dish and the external environment. So let's look at some of the characteristics of the non-vented Petri dish. 1 is it has a smooth and tightly fitting lid. And 2, little to no air exchange occurs between the inside and outside of the dish. And 3, it helps to retain moisture within the plate. And 4, it provides greater protection against contamination during microbial work. Advantages and disadvantages of the non-vented Petri dish. So let's start by looking at the advantages. So 1, it reduces the risk of contamination from airborne microorganisms. And 2, it prevents agar media from drying out quickly. And 3, it is suitable for long incubation period. And 4, it maintains a more stable internal environment for microbial growth. And for the disadvantages, the condensation may accumulate on the lid, making colony observation difficult. And also, limited air circulation may not be ideal for some strictly aerobic microorganisms. And finally, care must always be taken to avoid cross-contamination during incubation. And the last type of Petri dish is the vented Petri dish. And this is a type of Petri dish whose lid has small raised ribs or gaps around its edge. And these are the raised ribs or gaps around the edge of the Petri dish. And these vents basically is to allow a limited exchange of air between the inside of the dish and the external environment. So the characteristics of the vented Petri dish are: 1, it has small openings or vents in the lid that allows air circulation within the plate. And it also reduces the buildup of condensation, which are water droplets on the lid. And it's commonly used for culturing aerobic microorganisms that require oxygen. Advantages and the disadvantages of the vented Petri dish. So let's start by looking at the advantages. 1, it improves oxygen availability for microbial growth, and it also minimizes excess moisture and condensation, and it is suitable for routine microbiological culture work. And for the disadvantages, it slightly increases the risk of contamination as compared with the non-vented dish because air can enter through the vent. And the agar media used may dry out faster during long incubation period. So thanks for watching, and if you found this video helpful, please like, share and subscribe to Perfect Lab. Leave your comment or questions below, and don't forget to scan the code on your screen to follow us on WhatsApp for more laboratory updates and tutorials. See you in the next video.